This is the University of California, Davis, home of the Vector Genetics Laboratory in the School of Veterinary Medicine. The Vector Genetics Laboratory is led by Greg Lanzaro. His lab at UC Davis is one of 5 University of California campuses collaborating in the University of California Irvine Malaria Initiative, or UCIMI, a not-for-profit research collaborative whose mission is to contribute to malaria eradication from the planet. The team's work revolves around one particular mosquito, the Anopheles colutzii, the African malaria vector capable of transmitting malaria to humans. The UCIMI team has developed a genetically modified version of this mosquito using what's known as gene drive, which changes the DNA of the mosquito to prevent the parasite that transmits malaria from developing inside of it. As a result, the mosquito will not be capable of transmitting the deadly parasite to humans. An ambitious effort indeed, and one that could eliminate malaria from the planet. The UCIMI team has volunteered to be part of this demonstration. The team will be divided up into 2 groups. The first group consists of Greg himself and his colleague Anton Cornell, medical entomologist and professor for the Department of Entomology at UC Davis.
We believe that we have a solution that could contribute to the eradication of malaria in Africa.
We have to consult the community to be able to implement this genetically modified. It's a novel strategy and people need to know about it.
The second group consists of Anna Kormos, who leads the community engagement efforts for UCIMI, and Abram Estrada, UCIMI's research associate and resident mosquito handler for the lab.
This is an opportunity for us to show that we've tested it, we've tried it on ourselves, and it's safe.
What we're hoping to see is that there's actually no difference at all.
No one on the team has ever been bitten by the genetically modified mosquitoes, but they understand the importance of showing others that they are prepared to stand behind their work.
This demonstration will show that the UCIMI has full confidence in our modified mosquitoes, that it'll be an effective tool for malaria control without making major changes to the mosquito's biology or the ecosystem.
The team is joined by Dr. Robert Ditter, who will be measuring the size of the mosquito bites over time intervals that begin at the 30-minute mark, the 60-minute mark, the 4-hour mark, and the 24 and 48-hour marks.
Today's demonstration is designed to illustrate that the bite of the mosquitoes that have been genetically engineered by the UCIMI program, the bite is no different than being bitten by a normal wild-type mosquito.
The team has set up a space where participants will place their right forearm into a cage that contains wild-type female mosquitoes, while their left forearm goes into a cage that contains female genetically modified mosquitoes. The feeding on the left and right arm will occur simultaneously.
Work with them here. Okay, I'll be back.
The participants will leave their arms in the cage for 15 minutes, allowing the mosquitoes to feed. The mosquitoes have not been fed prior to the experiment to ensure our little friends have built up an appetite. As 2 p.m. approaches, Greg and Anton insert their arm into the cages and prepare for the first biting demonstration.
There we go. One of the concerns that a lot of people have with genetically modified mosquitoes is that are they going to be adversely affected? So that's an important question that I think we can address by demonstrating that if we allow ourselves to be bitten by non versus genetically modified mosquitoes, we'll show that there's no difference in the reactions that we get to their bites.
So these mosquitoes are quite avid. They're feeding on us nicely. With respect to the sensation, there seems to be no difference between the transgenic and the wild type. So the bites themselves don't feel any different. And of course, what we're interested in is to see if the reaction that we get from the bites are also the same, which we expect they will be.
At this point, Greg and Anton have had their arms inside the cages for a total of 15 minutes and will stand by as they prepare for their first measurement. Now, it's group number 2's turn to take part in the demonstration.
I volunteered for this demonstration because I believe in the technology, and I also believe that it is really important to address community concern about the safety of the modified mosquito. This is a brand new technology that has never been tested before, and what better way for us to show that it's safe and that we trust it than to expose ourselves to it. So I think that this is a really important demonstration for people who are living in areas where the modified mosquito is being considered for malaria control, so that they can see that it is safe.
As Anna and Abram's turn comes to an end, they will stand by for their first measurement.
Oh, God.
Shake your arm like that.
Oh, now I can see the welts that are arms out.
For the next 48 hours, Robert will identify and measure each bite on the participants to track its size and growth. He will do this at each time interval using a caliper, then compile the data when the demonstration is complete. The bites of each mosquito on each arm are not expected to differentiate widely for each individual person. It is a tedious process, but an important one, as the data is key to showing participating countries that the bite between each mosquito have no significant variance. The measurements continue throughout the day as Robert measures each bite carefully. Each participant is unique. Therefore, it is important to track the size of the bite at each time interval.
So we just wrapped up with the final measurements of the day, and we'll be back again tomorrow to do another reading at the 24-hour mark.
At the end of the day, the team debriefs and finishes up their day until tomorrow, when the 24-hour measure takes place. The team arrives over the next 2 days for their 24-hour and the 48-hour measurements.
Okay, so we are measuring the bites now at 24 hours at post feeding on the mosquitoes.
As Robert measures, the results reaffirm the team's hypothesis.
So we've completed our demonstration to look at the effect of biting between wild-type mosquitoes and those that have been genetically engineered. So we were interested in demonstrating that there's no difference between the bite of the two, which is what we expect because we know that the material that's been engineered into the mosquitoes is not present in the saliva. We allowed the mosquitoes to bite on us. We measured the size of the mosquito bites on the skin of 4 volunteers.
The important point is that we found no significant difference in individual responses to bites of wild-type and genetically engineered mosquitoes. It's normal to see that the response to the bite of mosquitoes vary from person to person. But for each person in this demonstration, the genetically engineered mosquito bite was no different from the bite of the wild-type mosquito.
We hopefully can reassure people living in communities where we're proposing to use these mosquitoes ultimately for malaria elimination.
The success of the demonstration is a step forward as the team prepares for its next visit to its partners in Africa, and one step closer the global effort to make malaria a thing of the past.